Good afternoon, everyone. I hope you can hear me clearly. Can you see the presentation slide?
Thank you. We can see the slides.
Okay, excellent. Thank you so much for the wonderful introduction. Yes, my name is Rufino. I am a PhD candidate at the University of the South Pacific. And today I'll be presenting to you on bridging the gaps in marine pollution data through innovative research methods for Pacific Island countries and territories. And before I begin, I'd like to acknowledge the sponsors of my research, the European Union, along with the Kingdom of Sweden, through the Pacific European Union Marine Partnership Program, in collaboration with the University of the South Pacific, who have made this research work possible. So the biggest problem that my project aims in tackling is pollutants. And a pollutant is basically contamination, substances or energy that results in or can result in adverse biological effects in animals and humans. And there's also an important distinction that I'd like to make with regards to the terms hazard and risk for the purpose of my presentation. So a hazard is something that has the potential to cause harm or danger, while a risk refers to the possibility or likelihood of that something to cause harm or danger when exposed. And some of the most common forms of pollutants, especially here in Fiji, that we have are things like oil spills, the discharge of untreated sewage water, and burning of rubbish, because our waste management system still has a lot more room for development, and that is needed. And every so often we see news articles related to pollution, so it would seem that the media is playing their role in public awareness very well. And we can say that we are aware of the extent of certain pollution-related issues, and that the government is not blind to a lot of those issues. But being aware and truly understanding the issue isn't really the same thing. In 2020, I published a review paper titled Knowledge Gaps in Ecotoxicology Studies of Marine Environments in Pacific Island Countries and Territories. And here we discussed the number of pollution-related scientific studies between 1975 and 2018 that was being conducted in Pacific Island countries and territories, and what the output contributions look like on a regional scale within Oceania. And as a whole, Oceania, that includes Australia and New Zealand, we looked at two of the largest scientific databases that indexes published studies, Scopus and Web of Science. And here we found that out of approximately 13,000 studies conducted in Oceania and indexed in Scopus, only 0.14% were studies out of Pacific Island countries or territories. And as for Web of Science, out of 30,000 pollution-related studies that were indexed, only 0.06% were studies out of Pacific Island countries and territories. And this is only on a regional scale, not on a global scale. So within Pacific Island countries and territories, only 9 island nations out of the 24 had peer-reviewed pollution studies, meaning that more than 60% of Pacific Island countries and territories had no baseline data for pollutants in a record of almost 45 years. And that was seriously concerning. And out of the 9 Pacific Island countries and territories that we looked at in here, you can see that had any indexed studies. Only Fiji had the most peer-reviewed studies, totaling 7. But even then, 7 peer-reviewed studies in 43 years, still worrying. And we found that only 4 types of pollutants were researched at all in Fiji. And all of these were done in almost a single type of environment. And none of the research in the Pacific Island countries and territories included measurements of the extent or the effects that pollutants had on marine animals. And Fiji, again, had the highest number of index studies of any of the Pacific Island countries that we looked at, totaling 7, and all done, again, in a single location and a habitat type, and lapsed over long periods of time. So there was a temporal gap as well, demonstrating geographical and time gaps in ecotoxicology studies in Pacific Island countries and territories. And so with the existing environmental risk assessments that are currently being conducted, what my research aims to do is to strengthen how we use environmental risk assessments to improve our conservation efforts, for example. And environmental risk assessments basically evaluates the characteristics of an environment by highlighting existing hazards of concern and its risks posed to human health due to its presence. And there are 2 main components to environmental risk assessments. The first one is a chemistry-based surveillance of the environment, where we quantify the levels of different pollutant classes from the soils or water samples or tissue samples of animals. And the second is a monitoring of the health itself of marine animals, which would translate into determining the quality of an environment. And to do this, we use biomarkers, which is what my research entails. And in Fiji, we have, and also in other Pacific Island countries and territories, our use of chemistry-based surveillance is wide, and that is a good thing. And that should definitely continue. But there is still, at the time of the research, there was still no biological monitoring that was done at any level on the environment. And we believe that by incorporating biological monitoring into environmental risk assessments for Pacific Island countries and territories, this would definitely help bridge many of the knowledge gaps around pollution. And the research process is, to explain it in a nutshell, in my research, I use early biomarker signals. So biomarkers can range from the molecular level all the way down to the ecosystem, and we measure the internal and external biomarkers. My research mainly looks at the internal biomarkers, so measuring stress responses in marine animal health, and seeing how that translates into the quality of the ecosystems as a whole. And so in this process, we look at, we think about and try to understand how, what are the possible ways a pollutant enters the body of a marine animal. In this case, let's consider a fish. And that can be through food intake or through respiration via its gills. And then we try to understand what happens inside of the body. And sometimes, in many cases, especially for organic chemical pollutants, the body tries to biotransform these chemicals, transforming it from one that is harmful to one that is harmless. And there are several processes that it undergoes, but primarily in 3 phases. And then we try to understand how these pollutants may be removed from the body. So it can be through excretion, it can be through egestion, or it can be through dilution. And finally, understanding what happens if the body is unable to completely flush out these pollutants from its body, or if there is retention, or if a chemical compound, the parent compound, is transformed into one that is more toxic or exerts higher levels of toxicity than the parent compound. There are several biomonitoring approaches or different biomarkers that have been utilized in Australia and in New Zealand, which we identified and we have recommended in the paper. And in my research, we looked at a few of these and tried to test its suitability and practicality, not just economics-wise, but also based on our environment type and our climatic conditions, being in a tropical environment. And so these range from physiology and morphology, again, biotransformation, which I had mentioned earlier, oxidative stress, neurotoxicity, endocrine disruption, and genotoxicity, so DNA integrity, etc. And last year, I published a paper that looked at multi-biomarker responses to polycyclic aromatic hydrocarbons and microplastics in the thumbprint emperor from a community, a local coastal community in Fiji that has a long-standing marine protected area, but also due to its close proximity to our capital, Suva, there are threats that exist, and we acknowledge and wanted to see whether, though, because many pollutants are transboundary, to see its effects on the thumbprint emperor, which we identified as a sentinel marine species. And so the marine protected area is a customary area that was established through the efforts of the community itself, not one that has been gazetted by the government. And it has been around for 20 years or so, and it covers around 19 square kilometers. You can see the community Vueti Navakavu, which is my study site. The community, there are 3 villages and 2 settlements that are part of Vueti Navakavu, locally managed marine area, or LMMA. And here is the city of Suva, the Suva Peninsula, where which is highly populated with several different types of industries across multiple types of sectors, all of which have a common outlet of wastewater disposal, including sewage water, which is around 200 nautical miles out from this area called Kinoya. And due to the southeast trade winds, we believe that many of these pollutants would be moving towards Vueti Navakavu locally managed marine area. And we wanted to investigate this further. We sampled a total of 53 matured thumbprint emperorfish, and we considered the 2 seasonalities in Fiji: the dry season, which is the wet season, November to April, and the dry season. And here are some of the data that we found for the mean total weight, the average total length, and the condition factor, which is a general reflection of the overall health of the fish. A high condition factor, a relatively high condition factor would mean the fish is in a generally good health. Low condition factor, the opposite. Similarly for the hepatosomatic index, which is a general measurement of stored energy. Basically, looking at how well the animal, in this case, the thumbprint emperor, is utilizing its feeding resources. And we did find a significant difference for the condition factor between the dry and wet season sample groups. But I will talk about this in a bit. And for the pollutants, for PAHs, or polycyclic aromatic hydrocarbons, for those of you who are unaware, these are some of the most common. This is one of the most common form of pollutants. It's released from the incomplete combustion of fossil fuels. So burning of rubbish or oil spills can all release PAHs into the environment. And it is also one of the key priority areas of concern for Pacific Island countries and territories who are committed to addressing the issue of hydrocarbons in our environment through the Pacific Regional Waste and Pollution Management Strategy. And we looked at the biliary PAH concentrations of 4 metabolites, 4 types of PAHs within the thumbprint emperor. These were benzoapyrene, 1-hydroxypyrene, phenanthrene, and naphthalene. And these metabolites give some reflection of the main local causes of pollution exposure. Mainly of these PAH metabolites, these are largely linked to pyrogenic and petrogenic sources. And I found that all bile samples, in all the fish samples that we collected, contain traces of the 4 PAHs. And while there was no significant differences which we found between the seasonal groups, it was clear that when compared to studies in other countries, the concentrations of each of these metabolites were relatively low. However, coastal communities and that tend to consume large quantities of fish, like Buetin avocavo, they could be at greater risk of health issues related to high PAH exposures. And there are certain metabolites, PAH metabolites like benzoapyrene, for example, which has been classified by the International Agency for Research on Cancer as being highly carcinogenic to humans. So in any case, these values identified, they present a baseline against which future research, future levels of biliary PAHs in the thumbprint emperor can be compared to, and continuous monitoring of biliary PAHs in the thumbprint emperor as an important local seafood fish can prove very helpful. And as for microplastics, so again, there is a growing concern of microplastics in Pacific Island countries, and this was highlighted by the Environmental Investigation Agency not too long ago. And marine plastics is now recognized as a priority to address in the 2050 strategy for Blue Pacific continent. And here we found that a total of 206 microplastics were found in the gastrointestinal systems of the thumbprint emperor from Buetin avocavo. And all of the fish samples contained at least 1 microplastic, which is a serious concern. 3 of the fish had up to 10 to 18 microplastics on average. And on average, the ingestion levels were not significantly higher in fish between the 2 seasonal groups. Majority of the microplastics found were less than 1 millimeter in size. So for environmental risk assessments, the size of microplastics, it may be a key factor in determining the range of animals that ingest them, as well as the retention rate. So for example, in one study of the gastrointestinal system of the Spinochromis, retention increased when microplastic sizes were reduced from 2 millimeters to 0.3 to 0.125 millimeters. And so investigating this further, can be very helpful for future studies in the region. And all microplastics were that were larger than 5 millimeters were classified into fragments, films, fibers, and microbeads based on their visual aspects. And we found that fibers in the wet season was the dominant form type, while in the dry season, a similar abundance of fibers and fragments were recorded. And so films overall had a lower abundance, but it was significantly higher statistically in the dry season than in the wet season. And microbeads had the lowest abundance and were found in the dry season only. Now, as for biomarkers, we looked at three main groups of biomarkers: the biotransformation enzymes, the enzyme activities to measure oxidative stress, and those that show biological damages. These include, for biotransformation, ethoxyresorufin O-deethylase activity, and that was phase 1 biotransformation. And for phase 2, we looked at glutathionase transferase enzyme activity. For oxidative stress, GST is also an antioxidant enzyme. So it was considered under oxidative stress as well, along with glutathione peroxidase and glutathione reductase activities. And for biological damages, we looked at lipid peroxidation and micronuclear assay, which is genotoxicity. And now we found that there were both phases of biotransformation were higher in the dry season compared to the wet season, though with no statistical significant difference. And on average, both components of biotransformation were relatively low compared with studies in other countries. And for oxidative stress responses, I measured the activities of, GSD, GPX, and GR enzymes, including the concentration of lipid peroxidation. And these levels were also relatively low when compared to studies from other countries. And for genotoxicity, the micronuclei assay, by counting the occurrences of abnormal nuclei formations in the red blood cells of the thumbprint emperor, we also found that these levels were very close between the seasonal sample groups, and on average was relatively low in levels when compared to studies in other countries. And while some of these biomarkers did not show statistical significant difference, there are biological relevant effects and efforts that should be carefully considered. Both PAHs and microplastics are relatively low, as well as the biochemical responses in the thumbprint emperor to those present levels. So as a baseline study for these biomonitoring parameters, it is important not to exclude the prospect of this community of wet in Avocavo having a 20 long year locally marine protected area as being of some consequence to the health of the thumbprint emperor. And again, in this publication of the preliminary studies, we do not assume a normal biological state being defined for the thumbprint emperor, and that it only serves as a reference point for future pollution studies in seafood fish in Fiji. So just to conclude, there is a need for more data for environmental risk assessments due to quantitative, geographic, and temporal pollutant type gaps for studies in Pacific Island countries and territories. And this includes a disparity in the selection of components for an effective environmental risk assessment, such as the absence of a biomonitoring or biological monitoring approach. And my PhD research is coming to an end, fingers crossed, but there is still more work to be conducted. And indeed, there's more space for researchers to build on this baseline research. So I hope this research will contribute to more pollution studies in Fiji and the region to help close existing knowledge gaps identified here. And without taking any more time, I will just show you this slide and conclude my presentation. And if you have any questions, please feel free to ask. Thank you.
Thank you very much, Rufino. We'll take some questions. My suggestion is, if it's a simple question, I can try and repeat it. If it's a complex one, I suggest come up and talk into the mic. So, questions.
Just for microplastics in general, I'd like to hear the experience on the field in Fiji, whether the microplastics plastic they find in the ocean, whether things is coming from Fiji or it's coming from other places.
So the question from Laurent is your experience with plastic pollution in Fiji, the microplastics and macroplastics, whether you think they're coming from Fiji or from beyond.
Well, I will just echo the findings from WWF Pacific, and according to their studies, majority of these marine plastics are coming from outside of Fiji. Yes, that's all I'll say right now. I really don't want to assume, but yes, that is one of the findings from WWF Pacific. The majority of these plastics are coming from outside of Fiji.
Are there any other species in addition to the thumbprint emperor that would be good to look into the impacts of these pollutants and use as a biomarker?
The question from Emma is, do you think there are any other species other than thumbprint emperor that could be a useful, biomarker species?
Yes, there's certainly many other sentinel species out there. And for my research, when I first started, Fiji had, none of the Pacific Island countries and territories had any sentinel marine species that were identified. And so we went through, I think, tested 20 species initially, and we settled with 3 species, and these were the thumbprint emperor, the flathead mullet, and a snapper fish, species of snapper. And there's also many shellfish as well that are really great sentinel species and bioindicators, which can be used. And indeed, New Zealand and Australia have several that have already been identified and can be used.
Slightly quite a basic question, but there was a difference in the types of microplastic found based on the season. But I was being much more predominant the wet season. What was the variation?
So the question from Hannah was, there was quite a variation, seasonal variation in the type of microplastics that you found. For example, lots of fiber in the wet season, I think it was. So what's the reason?
Yeah.
What was the reason that you concluded why that was the case?
We hypothesized that during the wet season, majority of the water that flows down gray water through our sewage and our sewage treatment plants, it's only primary and secondary level. We do not have a tertiary level. It does not go up to a tertiary level, and so it's difficult to filter out many of these types of very fine particulates, and so our hypothesis was that due to heavy precipitation, the sewage treatment plants, and the outfalls and drainage systems cause or are sources of these high levels of fibers.